Hi everyone, my name is Tudor Groza. I come from a land far away from here, from Australia, from Sydney, the King Kong Center of Clinical Genomics. And I should probably admit that the title is slightly misleading, and it probably is more interesting than the actual talk, but I'm hoping that I will still sort of, raise enough attention for you to perhaps go out there and then read the paper and then, get back in touch with us to discuss the several issues or several aspects we have covered. Just a sort of quick count of hands. How many of you have heard of your human phenotype ontology? Oh, good. So I can skip half of my presentation then, I guess. I will still, for probably the 2-3 people that did not raise their hand, I'm just going to give a very, very brief background in the human phenotype ontology. As you all know, it's one of the, let's say, one of the ontologies in the modeling phenotyping that has gained quite a lot of traction. It has 21 top-level categories, or it organizes knowledge by means of the organ systems and abnormality of different organ systems. It has, 11,000 classes and quite a number of annotations, 100,000 plus annotations for around 7,000 disorders, rare disorders. And you can see that the sort of annotations that the human phenotype ontology, has been used for are things like to describe, rare disorders by means of phenotypic characterization. So things like, Marfan syndrome is characterized by arachnodactyly or ectopia lentis. Now, so far, the human phenotype ontology, besides doing annotation and for text mining purposes, so on and so forth, has been used successfully in a couple of or various number of projects. One of them, or let's say two of them, are related to gene prioritization or variant prioritization. In this particular case, when we know that there are some gene disease associations, and we're trying to do variant prioritization, there's the Phoenix tool that goes and tries to combine the standard variant filtering approaches, the ones on the left, with a comparison of the phenotypic profile of the patients with existing knowledge or the body of knowledge, both in human data as well as in the model organism, to try to come up with a combined score for the final variant. Similarly, it has also been used for discovering new gene disease associations, when we know that we have a patient with a phenotypic profile for a patient, and we are able to infer by means of the semantic similarity of that phenotypic profile of the patient with, for example, with a model organism, perhaps a gene that may be of interest in that particular patient. And I know that this is not necessarily the best picture to show this, but the idea would be that you find the semantic similarity between the profiles, and you find that that particular mouse model has a variant in KIF1A, and then the idea would be then for you to turn your attention towards KIF1A in the case of the patient, and has been used successfully to diagnose, as far as I know, 7 patient cases in the UDP, so undiagnosed disease program. Now, the purpose of this talk is to see to what extent HPO, as we know it now, and again, keeping in mind the fact that it has been built and has been used so far for rare disorders, can be applied to common diseases or complex diseases, right? I'm just going to scratch the surface with respect to the work that we've been done and we've been doing, and kindly ask you to perhaps have a look at our paper and download the data and play with the data, right? So this is more or less the sort of the process we went through to understand to what extent HPO is useful for some aspects related to common disorders. So we started from the Medline corpus, and we did automatic concept recognition, obviously because it was far too much to do manual annotation on that. And then we extracted HPO terms out of the Medline corpus, and then we did a bit of concept ranking and filtering, because obviously there's a lot of noise, both because of the concept recognizer not being perfect, as well as because in general the sort of associations that you may find from the data are not the ones that you would be looking for. And then we try to understand to what extent, for example, the phenotypic profile of the common diseases, or what it would be, let's say, the genetic background of common disease by means of their phenotypic overlap, as well as the genetic background of the rare and common diseases, again, by means of the phenotypic overlap. And in this presentation, I will cover only a couple of things that we've done, and again, I invite you to read the paper for everything. So we try to understand, if we can recreate a common disease network using the disease ontology, as well as to understand the genotypic and phenotypic overlap between common diseases, as well as the phenotypic and genotypic overlap between common and rare disorders. So I'm going to go through each of these steps very briefly. Again, I'm just scratching the surface. I won't go into details. I'm more than glad to answer questions afterwards. So concept recognition and filtering. Again, with you, obviously, I'm assuming that the word or the concept of concept recognition is quite familiar to you. So what we did was to mine HPO terms out of abstracts, of Medline abstracts. We used the 2014 Medline corpus, around 22 million articles. Now, one of the things that is quite important to mention at this point is that we've taken for granted the fact that we have mesh annotations in those, or mesh metadata in those abstracts. And what we did was only to focus on the category C, on the diseases, right? From those sort of categories, coming back to one of the talks that was yesterday, we filtered out manually, and this is by means of a lot of work being put by Peter Robinson, and we filtered out only those that are unambiguously defined as diseases. So we filtered out terms that may be both characterized as diseases as well as phenotypes, and we left only those that, at least from a clinical perspective, they seem to be only diseases and not also phenotypes. So we ended up with around 3,000 entries, and by means of the first pass of the concept recognizer over this corpus, and after the mesh filtering, we ended up with around 1 million HPO annotations for these 3,000 diseases. Because again, obviously the concept recognizer is far from perfect, and we don't really infer proper relations from text, but simple co-occurrence between the mesh and the HPO terms. We had to do a bit of ranking and filtering, so to trim out the noise, and we did again. There's an algorithm there, but just to sort of briefly summarize it, we did an initial ranking using. Actually, that's IDF, I'm sorry, third frequency inverse document frequency. And then what we did, we tried to incrementally cluster terms based on the high top level HPO category with the assumption that probably terms that fit in the same category, such a growth abnormality, would cluster more together, and then they would better define, let's say, one particular disorder of interest. At the end of this process, we ended up with around 130K HPO annotations, with a coverage of more or less half of HPO, and with an average of around 41 annotations per mesh disease. And you can see here, in principle, the distribution of the type of phenotypes that we've covered in this data set. Now we've done that again. So far, everything was automatic. Then we tried to understand to what extent the current results are valid. So we performed a selective manual validation where we picked 41 random diseases from 13 disease ontology categories. I'll go to disease ontology in a second. And we had basically a team of curators that went through them and tried to see which annotations we found are correct and which are incorrect, but as well as trying to understand what we have missed, right? So not only just try to say, okay, yes, this is correct, this is incorrect, but also to add annotations that we've missed in order for us to be able to compute a proper F-score. So you can see there that the selective performance, which basically means that we've just used really this small set, where we ended up with a 66% precision, and in some cases we've actually hit some better numbers in different DO categories, such as the disease by infectious agent and, for example, immune system diseases. Out of those, 41 random diseases, 82% precision. Now, what we did then was to use this manual validation process as a way to learn basically the parameters of the ranking and filtering algorithm in order to maximize one of the three components, obviously, of the efficiency score, and we ended up, going for F-score in order to do have a proper balance between precision and recall. Well, at the end of the day, what we did was the final result was basically to settle on an F-score of 45% overall. Over the entire set of diseases, overall the entire set of annotations. Okay, now so this was then basically the final, let's say, corpus of annotation we've used, right, in order to go to the next steps and do the actual studies. And I'm going to show you two very small studies we've done. One is the Common Disease Network. So this is, I was saying that I'm coming back to the disease ontology. Disease ontology has, classified diseases in different categories. You can see there are 13 categories ranging from nervous system, respiratory system disease, and so on and so forth. They were, in principle, using basically the mesh terms that we've focused on from 3,175 mesh terms. We ended up using 1,600 disease ontology terms, using the mesh to disease ontology mapping kindly provided by Lynn and the colleagues, and as well as manually created by us. And what we try to do is to really try to understand whether just by using the phenotypes that we've mined and filtered, if we can recreate to some extent of these categories, the top level categories in the disease ontology. The way in which constructed the network, again, very briefly, we did we used the information content of the terms and to compute the pairwise similarity and to cluster them, and with this pairwise similarity was symmetric. Now this is the outcome. Quite a pretty picture. I love network pictures because usually they're so colorful. So if we are to dig into, obviously the results were far from perfect, right? But there were some interesting insights we were able to come up with. So for example, you can see the cardiovascular system was quite well recreated, or at least in part. So basically that red cluster over there. The diseases of cellular proliferation also, they are fairly well recreated. So that's sort of that category. But then it sort of stopped there in terms of the higher level clustering. Nevertheless, it's interesting to see that we were able to recreate subclusters of diseases, and not necessarily the way in which they were initially classified at the top level by DO, but really at around the particular organ system, let's say. And you can see here we had a couple of the mouth diseases are fairly well clustered. We had the eye diseases fairly well clustered, and that's basically it. Again, I would invite you to have a look at the paper. We also did additional studies to try to validate the network that we've created there. The second study I'm going to show you is the idea of phenotypic and genotypic overlap of the common diseases. So we've used GWAS Central, where a subset of GWAS, that's GWAS Central data, just to make it clear, where we've used the RSNPs to mesh terms association that's being provided by the data set. And then we also use the ScanDB for the RSNPs to map gene column in order to get from mesh terms basically to genes, right? And the way in which we've done the process, again, we had a mapping and filtering process. We started with around 16K unique SNPs, which were associated, around 800 around 800 of them being associated with more than one disease, and in total there were 300 such unique diseases. Again, in the context of those 3,000 mesh terms that we have focused on, and by doing manual filtering and direct mapping between sort of the data that we had and this data from the GWAS Center, we ended up with around 500 unique SNPs and 79 unique diseases. Now, the way in which then we try to again cluster them and to try to understand the phenotypic and genotypic overlap was by doing pairwise comparison, and we used adapted Jacquard index because again we're using ontologies, and therefore we thought that we can actually use the ontology structure to go beyond just a simple set intersection and union. Again, I'm not going to into details, but the point is that we're using also the parents and the sort of the children of the particular disorder to compute the Jacquard index. And what we found was that we were able to reach 0.25, Jacquard index plus minus 1.3, and which was more or less double than if you were to do this in a randomized fashion. Now, the interesting thing was that then when we had a bit of a look at the data, you were able to find such sort of dense clusters of RS SNPs to phenotype terms. In this particular case, you can see such a network of autoimmune phenotypes, which are known to be characterizing things like the Sjögren syndrome or the systemic lupus erythematosus. Okay, now. These are sort of very briefly the two studies we've done. Now, another interesting outcome from this study was a PubMed browser. So obviously, having the data, we were thinking, why not just putting it out there and enable people to use it, not only by really using the data, but also sort of browsing the publications by means of the data. And you can see here, we've created this very small PubMed browser that allows you to search for a common disease, and you can very quickly filter using the HPO terms, and there's also a mesh disease down there somewhere list. And the idea here is that basically you can very quickly get a glimpse of the HPO terms characterizing the disease that you are looking at. Okay, not only to get to the abstracts, but also to see that, for example, in this particular example, you have the Parkinson disease, which is characterized by dyskinesia, depression, and so on and so forth, or at least there are terms that occur often with it. And then when you go to the actual Parkinson disease, the actual mesh term, what you get is, besides all the abstract, you get the ranked list of phenotype terms based on that ranking and filtering process we've done. The data, so the actual PubMed browser is out there. The data is freely available. Please download it, use it. Let us know if you find issues with it. So in terms of conclusions, obviously there's quite a number of limitations to the study that we've done. On the text mining, there's both, we had plenty of both false positive as well as false negative, especially false negatives. And obviously, the mapping and filtering of mesh terms, because it was driven by, human interpretation, and because of this idea of ambiguity between a disorder and a phenotype, it's obviously also subjective. But as outcomes, we can claim that we've created a corpus of around 3,000 disorders annotated now with HPO concepts, so 3,000 complex or common disorders annotated with HPO concepts, and we have presented, and again, please, I refer to the paper. We have presented a couple of interesting examples of how to use this data employing HPO for understanding, the genetic background of common disorders as well as the overlap with common and rare diseases. So a lot of acknowledgements. So the Monarch initiatives and different other people that have been we're working together as part of a team in these experiments. And I would kindly refer you to the paper that was just been published and the PubMed browser, which is down there. Thank you so much.
Thank you very much. Are there questions in the audience? Yes?
Are you planning to integrate these results? Because I think that they are not integrated in the current version of the new Ontology, because they are from text mining. Are you planning to?
So the next step is basically to manually curate the data, and yes, it will be part of the Monarch initiative, the part of the entire process. Sorry.
Other questions? I have one. So that's the snapshot from last year, 2014.
Yeah. Yes.
So how do you plan to update this now that the paper is published?
Well, it's a good question. So firstly, we're trying to rerunning the study. We've improved the concept recognizing. We ran a study on a 2015 corpus, but then the actual incremental updating will be a bit of a challenge.
Okay. And did you actually look at all comorbidities? So really like looking at the sets of terms that are more frequently found, or disease that were more frequently found into your disease network, or have you not looked into this for the comorbidities? Because fundamentally, we don't have a single disease. We are a composite of several dozens, if not higher number of diseases. So have you looked at all at the comorbidity, or are you planning to look at it?
We were quite selective. Obviously, we're trying to find some good examples in this particular case. But this is basically the start. We're hoping that it's the start of a much longer project that will go in that direction as well.
Thank you very much.
Thanks again.
Thanks again. So while we have the next presenter that is giving